In the course of his study in the late 1990s, we made two absolutely fascinating observations that were challenging two huge dogmas of traditional science. So let's talk about it. Hello, I am Christian Drapeau. I'm a stem cell scientist and herbalist, and I help people live longer, better lives. So this was the mid-1990s where something was emerging at that time called live blood microscopy. So you take a drop of blood from somebody, you put that on a microscopic slide, and you look at them under the microscope using either phase contrast, natural light, natural density, or phase contrast. And as you look at these different modalities of microscopy, then you can observe different things in the blood, and you can use this kinds of observations to diagnose people with their health, identifying some problems that they may have. And I thought at the time that it would be absolutely amazing if we could corroborate, verify the type of diagnosis that were made with this approach, blood microscopy, with traditional means of diagnosis. If you do blood microscopy and what you see leads you to say, oh, this person has liver problems or lung problems, heart problems, well, let's do traditional diagnosis to see if we can corroborate that kind of observation. And unfortunately, I'm not saying here that live blood microscopy has no use or is not relevant, but we could not corroborate those kinds of observations. So although the work was very interesting, we had to drop this whole project the aim here was to use blood microscopy as a tool to be able to do a study in a population. You take a population, you give them a dietary supplement, an herbal extract that you want to study, and the effect can be different in different people. So by using one method, blood microscopy, it would allow us to basically see all the different benefits that we can get in a population. That was the idea. And we could not do it. So we dropped this project, but as part of this project, we came across a number of individuals, people with chronic fatigue, fibromyalgia, and Lyme's disease, in which we made a very, very bizarre observation. In the blood samples from these people, we would see under the microscope bizarre things that were actually moving, like small, tiny parasites, like spirochete-like microorganism that had motility, like we would see them spiral and actually like swimming in the blood, in the plasma. So we tried to understand where these were coming from, and at some point, as we extracted and isolated the red blood cells of these individuals and resuspended them into artificial plasma, if we would change the temperature or the pH, we would see these boogers, we used to call them, popping out of the red blood cells and starting to move around. So they were actually living in red blood cells in these individuals, and then upon certain conditions would then leave the red blood cells and go into the plasma. And around the same time, there is a scientist at McGill University that had made exactly the same observation. He had taken pictures of these microorganisms not only coming out of red blood cells, but also changing forms. And this is one of the dogmas of science that was challenged, by these observations, is that these microorganisms looked in certain condition like a cocci. Then under certain other conditions, that same microorganism would transform into a bacillus, and then at some point would disappear, and then you would turn the microscope into phase contrast and you would see that it was still there, but now it had changed its membrane structure and was now behaving like a fungal organism. So the question was, it seemed like it was an organism that was crossing species, and we know in science species is something that is determined, fixed, and cannot change. So here we had an observation, again not made by us. We had the same observation, but it was published by somebody at McGill University documenting the same kind of observation. So we had this microorganism that had been observed to come out from red blood cells, and interestingly, if we would take these red blood cells and put and suspend them in the plasma of a healthy person, then these microorganisms would not come out of these red blood cells. So it seemed like there was an immune factor in the plasma of healthy people that would keep these microorganisms in check. This fellow at McGill had suggested in the paper that he had published, had suggested that it appeared to him, at least, that these microorganisms were living in symbiosis within the red blood cells. potentially playing a role in the maintenance or the incorporation of iron in hemoglobin. It was not clear. But to him, there was probably some sort of symbiotic relationship, or at the very least, that's the place in the body where these microorganisms seem to be wanting to live. So in any case, when we blended them, the red blood cells with the plasma of healthy people, we did not see them. And incidentally, why all of this was linked with our work with AFA is that it was all part of an investigation that we were doing with this blue-green algae from Climate Lake called Aphanizomenon flos-aquae, in short AFA, is that people consuming AFA, we would not see these microorganisms. So at some point we made an extract of AFA and we simply mixed that AFA extract with the plasma of these individuals, and it would also keep these microorganisms in check. But the one thing that was also quite fascinating with this is that at some point, and I have a video of this somewhere, so I will try to find it back, but it was something absolutely amazing. When you look at these microorganisms, or you look at anything liquid under a microscope, and so you put the cover slide, and the edge of the cover slide dries, and it basically fixes the liquid under that cover slide, so you can look at everything that is in that liquid. Well, sometimes the dried part opens, so the seal opens, and it creates evaporation, so it creates a flow under the slide. So that happens, and I happened to be filming at that time. So you start to see a flow under the microscope, and a number of these microorganisms started to be carried by that flow. So they started to kind of swim counter current, and they were carried by the current. So they suddenly all ganged together. There was probably, I don't know, like a dozen of them. They all grouped together, and together formed a front, and they started to swim counter current. So my thought was that, okay, we're looking at, so first we talked about one organism crossing species. And now we had an overall condition where we were identifying microorganisms living in the blood. We all know the blood is sterile. What we don't oftentimes understand is that the blood is not sterile. It's just that on the normal growing media on which we normally assess whether the blood contains bacteria, whatever bacteria is in the blood, they're not growing on those media. So the conclusion is that there's nothing growing from your blood, but there are ways of growing these microorganisms. So what we found is that if we were to take red blood cells, spin them with a G-force high enough to rupture the red blood cells, collect the inside of these red blood cells, we were able to grow these microorganisms in that kind of environment. So there are microorganisms in that blood, at least there were microorganisms in the blood of these individuals that had chronic fatigue, Lyme's, or fibromyalgia, and these microorganisms were kept in check if mixed or blended or suspended with plasma from healthy people, or putting some AFA in the plasma, and we did not see those in people consuming AFA. These microorganisms therefore were living in the blood, so another challenge here of one of the dogmas of traditional science, that the blood is sterile. And the third one is that these boogers seem to have, as bizarre as it seems, it's almost hard to say it, but they seem to have some sort of social behavior. They group together in order to aim at one specific movement together. I will dig for that video. Hopefully I can find it. But in any case, all of this to say, as we were doing this work on microscopy, it led to a number of observations basically telling us that there appeared to be a microorganism in the blood of individuals with chronic fatigue, Lyme's disease, and fibromyalgia. It is possible to keep that microorganism in check with AFA, and interestingly enough, historically, we have seen many cases of people consuming blue-green algae and reporting benefits with chronic fatigue, fibromyalgia, and Lyme's disease. So if you have any of these conditions, then give it a try. Try it yourself and see what it can do for you. Climate Lake blue-green algae.